this video, let us try to understand various facets of drug discovery program. A drug discovery program initiates because there is a disease or a clinical condition without suitable medical products available in the market, and it is the unmet clinical need which is the underlying driving motivation for the project. The initial research often in some laboratories in the academic, and it can generate or develop some hypothesis so that can act via some of the protein mechanism such that it can show some therapeutical effect in the disease. So now coming with the validation of the process, it's not properly justified. So during lead discovery, an initiative search ensures finding of a drug like a small molecule or some biochemical or therapeutical active substance, so which typically it will be termed in a development candidate, and further it may progress into preclinical studies, and sometimes a successful result we may come across if it is processed towards the clinical trials. So the drug discovery and development is mainly concerned with enhancing the property of the drugs that are in the clinical use and developing new ways of treating medical conditions, and getting a drug into the market generally takes a years of effort, like decades, and involves many individuals with different expertization from a broader range of various disciplines. They work together, and at the end some tested molecules may pass out from the new drug discovery. Let us see the various main stages in the modern drug discovery. We can enlist into various medical or chemist, so like in the various 4 to 6 stages. The first, it may be the selecting the medical condition to be treated, followed by identification of the drug target, finding suitable bioassays, identification of lead compounds, examination of structure activity relationship of the lead molecules, drug design and optimization, manufacturing process, designing and applying for patent or clinical trials, regulatory approach, then release of the drug to the market, and also it continues with post market surveillance. These are the various things what you can come across. First, it may start with the understanding the disease condition. Let us continue the discussion with the target identification. So many times drug fail in the clinic for 2 reasons. The first is that they do not work, and the second is that they are not safe. As such, one of the most important steps in developing a new drug is target identification and validation. A target is a broad term which can be applied to a range of biological molecules, which may include for drug action. For example, it may be proteins, genes, and even RNA. A good target need to be effective, safe, and meet clinical and commercial needs, and many times all the things what we are enlisted may be required of, such that our drug can bound on the target and it can show some response. So many times the biologicals may be a target for the drugs. With that, we are going to measure both in vitro and in vivo activity. Say for example, G protein coupled receptors, very small molecular system which may be a target in the drug discovery process with many of the drugs. Whereas in some conditions like antibodies are good at blocking some of the protein or protein interactions, or sometimes good target identification and validation may also sometimes enables us to increase confidence in the relationship between the target and the disease, and also allows us to explore whether target modulation may require, which can lead to mechanism based with some unwanted or some side effects. So now coming with the lead identification. During the early stage of drug discovery for a certain diseases, some of the mechanism behind that disease would be studied in detail. So these studies will include identification of the cellular and genetic factors involved in the disease, followed by the identification of potential targets in order to ensure that the biological target is involved in disease, where some in vitro studies like isolated cells and in vivo like animal models testings can also be performed. This is also known as a target validation stage. Modern target validation may involves a combination of in vivo and in vitro, or sometimes like in silico, which prefers usage of computers, may also be employed in order to study the drug action. The results of target validation stage can assist us in lead compound identification. So lead compounds means what? So these lead compounds are chemical compounds that show desirable biological or pharmacological activity and may initiate the development of new chemically relevant compound. These lead compounds are typically used as starting points in drug design to give new drug entities. So these drug design strategies can be used to improve the compound's pharmacodynamics and also some pharmacokinetic properties of the lead compounds. So these resulting compounds from drug design may go through a series of preclinical studies and become a clinical candidate if the compound does not exhibit adverse effects or toxicity during in vivo and in vitro studies. So after completion of this long process of studies, the molecule may go to the market. So there also post-marketing surveillance can be done. It's a part of the clinical trials. The compounds that passes are released on the market. So they may be new drug and it may be studied of. These new drugs are monitored for its safety after their release in the market. So this process is called as a post-marketing surveillance or phase 4 clinical trials. So now let us see what are the possible lead sources or the sources of lead molecules. Like we have natural products or sometimes like chemical libraries, what we have discussed in the initial introduction. From that we may get our componential medicinal chemistry may also provide the lead molecules. Now coming with these natural products, so most drugs in the clinical trials use the natural products or derivatives and analogs of some natural products. So many of the natural products are used as a lead molecules. A vast amount of natural products so contain some structural features that are very much difficult to achieve through the synthetic organic chemistry. The structural complexity of many of the natural products makes the developers to make it a simple molecule by slight structural modification. So many of the natural products you can see in the molecules like plant-rich sources which are used in the pharmaceuticals. Say for example antimicrobial agent quinine, which is isolated from the bark of cinchona trees. So let us see about the chemical libraries. Chemical libraries are nothing but a large collection of synthetic compounds. Chemical libraries are generated through the techniques such as combinatorial chemistries. These combinatorial chemistries which having a method used to generate libraries of compounds with sizes range from hundreds to millions in a single process. So once that new lead molecules is identified, the chemistry or the chemical structure will be studied of. A lead molecule once it is found in the chemical libraries, so it can be used for searching possible leads through high throughput screening method, what we are told as HTS methods. So thereby whatever the database you are going to get, so that will be get stored with the help of computer process. Then coming with the lead optimization phase, the object of the final drug discovery phase is maintained for favorable properties in lead compounds. So where we will be improving any deficiencies if you are observed in the lead structure. So once the compound have reached this stage, may have met the initial goals of the lead optimization phases, and it's ready for some final characterization before it is declared for preclinical studies. So now this process involves exploration of the synthetically product in order to produce some potential backup of the molecules. So in case compounds, if it is undergoing further preclinical or clinical characterization fails, and more oftenly like we may look up for some follow-up series. So however, in general, these molecules need to be examined in various models for its genotoxicity by using various tests like Ames test can be done, and some in vivo models can be used for studying some general behavior of the molecules. Even a high dose pharmacology, pharmacokinetics and dynamics, and even dose related studies and pharmacokinetics activities, drug induced metabolism, and even the metabolic profiling can also be conducted. Then coming with the last aspect, that is the clinical testing, where we will call it as screening and designing. So this is a process of finding a new drug against a chosen target. This target in the initial steps only we are identified. For a particular disease, usually involves HTS technique, where in large libraries of chemicals are tested to the ability to modify the target. For example, if the target is a G protein coupled receptor, compounds will be screened for ability to inhibit or stimulate the receptors. If the target is a protein kinase, the chemical will be tested for their ability to inhibit that kinase. So another important function of this HTS is to show how selective the compounds are for the chosen target. As one wants to find a molecules which will interfere with only the chosen target or not, that can be studied by using this HTS technique. But coming with the conclusion point of this, it is unlikely that a perfect drug candidate will be emerged from these early screening runs. One of the steps is to screen for a compound that are unlikely to develop into drugs. For example, compounds that are hit in almost every assays, which passes the assays, can be classified by medicinal chemists as a pan-assay interfere molecules and are removed at this stage if they are not already removed from the chemical library. So with the attempt in this test, so we are going to study about some features of the lead molecule, like increased activity against the chosen target, or reduced activity in unrelated target, or improved drug like action or ADME properties of the molecules. With the help of this process, so we will be keep on screening the drug for its activity by using various in vitro and in vivo molecules.